In this video, we're talking about viral components. So I'm Dr. Poe, and this is for BIOS242. And so our goals here are to describe the molecular components of viruses, describe the various ways scientists classify viruses. A lot of them are based on the molecular components. And then, of course, to compare the morphology and structure of a virus to a living cell. So viruses are really important to learn about because they can infect every type of cell. They can attack bacteria, and so we call those bacteriophage. They can attack plants and cause harm to our harvests. They can attack fungi. They can attack protozoa. They can attack, and of course, humans as hosts as well. So everywhere that a virus is, it is an acellular pathogen. These are particles, not organisms. So there is some debate about whether or not we can classify them as alive based on the current definition of living being you have to have a cell and you have to be able to replicate. Well, viruses do not have cells. They are acellular. They are extremely small because they have to be able to enter into cells. We call them particles. And the first viruses were actually discovered in plants, tobacco mosaic virus. And so there was something causing these issues, and so they filtered out all these different things, and each type of filter caught the cells, but viruses are so small, they fit through those teeny tiny little holes in the filters, and they were still causing damage. So these are considered filterable particles. So it's really important to kind of figure out what they're made of, because if we know what they're made of, then we can really understand how to stop them. So the viral size range is actually quite extensive, but they are all consistently much smaller than cells. We can view viruses by using scanning electron microscopy and transmission electron microscopy. So the scanning electron microscopy, that'll help us view the surface, and transmission electron microscopy will help us to view inside. And by using this information, we were able to actually visualize viruses for the first time and view their molecular components. When this happened, we realized that viruses do not look like cells at all. So they do not have any ribosomes. They have no intercellular components. They have no cytoplasm. They have no organelles. It's nuts. So what do they have? What they have are two main parts that are common to every single virus, and that is an external coating, and that coating is made of protein, and it's called the capsid. And then they also have some core containing some nucleic acid, either DNA or RNA, never both. And it gets a little crazy because in viruses, we know in our cells, we normally have double-stranded DNA. In viruses, we can have single-stranded DNA or double-stranded DNA, which is very unique. And then for RNA, we know in our cells it's normally single-stranded, and we see that as well. Or it could also be double-stranded RNA there. Also, in terms of RNA, it could be the positive sense RNA, and if it's positive sense, then I'm positive it can go straight in to making protein. And if it's the minus sense RNA, that has to be turned first into the plus sense, so we'd have to do some complementary base pairing in order to get the plus sense RNA, and then from there we can get the protein. So that's something to keep in mind. Plus sense, I'm sure that's going to work. Minus sense, it has an extra step in there. So we see a lot of variation in viruses that way, and so one of the ways that we can classify them is by the type of nucleic acid. acid that they have. Is it single-stranded DNA? Is it double-stranded DNA? So we have groups of DNA viruses and we have groups of RNA viruses. In terms of the external coating, so the capsid, that's the shell that surrounds it. And then, so I'm just going to draw it kind of here. So this could be the capsid that's made of the protein coat. And inside we've got either DNA or RNA, not both. In our cells we have both, and our DNA is double-stranded and our RNA is single-stranded. Thank you very much. And viruses, it could be anything. Double-stranded, single-stranded, RNA, DNA, positive sense, negative sense. There's a lot of variety there. So if the virus is what's considered naked, this is all we see. It's this whole piece. It's just, this is the minimum. It's called the naked virus. I'm sure you'll remember that. Naked virus. They pretty much just have nucleic acid in the capsid. Sometimes that's called the nucleocapsid. And so if it's an enveloped virus, we've still got that capsid, we've still got some nucleic acid in there, whatever, what have you. And if it's an enveloped virus, it's going to have an extra coating. A lot of times, this is part of the host's cell membrane. So the virus kind of picks this up on its way out. And so this is what an enveloped virus would look like. So the DNA may or may not have, the nucleic acid at the core may or may not have information to encode this envelope. A lot of times it's from the host. Envelope, and it's not found in all viruses. Okay, so in addition to that, another component are these spikes. So these spikes can be used, and it could be on enveloped or on naked. And these can either project from the capsid itself or from the envelope itself. And really what these spikes do, the spikes are for recognition. The spikes help the host, help recognize the host, where it's supposed to go, those particular cells. Host cell. And then also it allows attachment there. Okay, and important vocab part here that I want to bring up is this one, virion. So what the virion is, that is the complete, ready-to-go virus particle that's ready to cause infection. Ready to go. So there's different phases in replication that are still considered viruses because they're still doing virus things, but the virion is the complete ready-to-go virus that has all of its components. If it has spikes, it's got spikes. If it's got an envelope, it's got an envelope. Those types of things. Okay, so in terms of capsids, bacterial viruses, which we call phage, so those are specific to bacterial cells. We'll call them virions. They kind of have really unique capsids. They kind of look alien to me. So they look like this, and then they've got kind of these specialized tubes and specialized attachments there, and basically, and they've still got, oops, I was using blue for nucleic acid, pardon me. Got some nucleic acid there. So the reason for this, and they could be symmetrical or not, etc. The reason for this particular shape is because these are going to sit on the outside of the host membrane and actually physically inject their DNA, or sorry, not DNA, it doesn't have to be DNA. I keep saying DNA, but it could be any nucleic acid into the host cell so that outside the capsid remains. The capsid that it came from is going to be empty and outside. And then that virus on the inside, this phage DNA, is then going to go steal the bacterial machinery and make the heads and make the tubes and make the legs. And then eventually those will all get reassembled back. And we'll talk in a little bit more detail about that. So bacterial viruses do this, animal viruses, it's very similar. And then from there, they can exit the cell. Okay, so that's kind of what bacteriophages kind of look like and what they kind of do. And a lot of times, enveloped viruses, so they got that lovely envelope on the outside, and maybe they come up to a host cell. I'm just going to draw the capsid in there and the nucleic acid in the same color. What they kind of do is they'll kind of fuse with the host cell a little bit, and then end up opening. So this is the original membrane, and there. And then they can inject their capsids and nucleic acids that way. So those are kind of the structural differences. They kind of have different mechanisms for replication and attachment, but really these spikes on the outside are going to recognize the host and are going to be specific to the host because viruses have lots of specificity, and these little legs, they can have specificity as well. Okay, so occasionally viruses also carry additional components that are not really found in host cells. And they do take some things from the host, some components from the host. They're going to use the DNA and RNA polymerases. And then remembering those that are enzymes that make the polymers RNA and DNA, and they'll also steal those ribosomes to make those protein coats. And some viruses are going to carry with them special enzymes like reverse transcriptase. So that's something our cells do not have. And what reverse transcriptase does is, if you remember the central dogma, which is DNA is transcribed to mRNA and translated to protein. So this is all based on base pairing, transcription, translation. Reverse transcription would actually be taking mRNA and making DNA. So this is really important for some of those, particularly important for retroviruses. So retro means backwards, so it's instead of going the forwards normal cell dogma direction, it's going backwards here. A really well-known retrovirus is HIV, human immunodeficiency virus. And so this virus packs with it, this is an animal virus, it packs with it a retrovirus that's able to take the encoded RNA that's found in the capsid and make DNA from that. And once that DNA is made, then we can follow the normal transcription translation using our host cell machinery. But it would not work without this reverse transcriptase enzyme that some viruses carry when they need it. Other things, if they're carrying RNA, they could also take other enzymes called replicase that will make make RNA copies so that it can make its RNA nucleic acid as much as needed. So they need special enzymes in some cases if our cells do not have those enzymes that can do those functions. Like we don't have reverse transcriptase, we don't have replicase. And if you need the RNA to be the complete virion, then you're going to have to bring those enzymes with you. Okay, so let's talk about, so those are the structures that you should definitely be aware of. Molecular components of viruses, we talked about capsids, we talked about nucleic acids, envelopes, spikes, nucleocaspids, so occasionally reverse transcriptase and other types of enzymes. Just describing the ways that scientists classify viruses is our next goal. So for classification, we really look at a couple main things, which I already mentioned. I already mentioned one of them at least. So got to look at the type of nucleic acid, because that tells us a lot. The DNA of viruses are going to replicate mostly in the nucleus because that's where the DNA is. The RNA viruses are going to replicate mostly in the cytoplasm because that's where the RNA is. If it's a retrovirus, that tells us something. If it's got positive sense RNA, negative sense RNA, single-stranded, double-stranded, etc., etc., etc. Those are really important types of classifications. Another type would be the host. Is this bacterial, in which case it's a phage? Does it affect plants? Does it affect animals? That's another way that we can classify them. And then shapes. So is it a helical capsid? Mostly for capsids here. Is it helical capsids? Is it even? Is it symmetrical? Is it multi-faced? All those types of things. So this has been used a lot of times. This is really sort of an informal classification scheme. And for more formal, we can go farther and look at like the DNA sequences and look at similarities in genetic makeup. So specific genetics, not just what type of nucleic acid it is, but what's the sequence, as well as structures and specific chemical compositions. So like, does the spike contain this glycoprotein? Does it contain that glycoprotein? So those are also ways that's for a more formal, especially in anything involving DNA genetics. That's going to make it extremely formal because it's very reliable. Doesn't change so much. Okay, and so from this, we don't have like the same classification system. These don't have a domain because they are not cells, and our domains are not prokaryotes. The bacteria, archaea, and eukarya, that's all based on organelles and cell types. We don't have cells here, so we have different classification there. So you can tell if something is a viral family if it ends in viridae. So if it's something viridae, you know it's a virus. And genera, that's a plural of genus, it's going to end in virus, and that's going to be a dead giveaway that something's a virus. So something like the human herpes virus, that's going to be in the family herpes viridae. And then the name is human herpes virus, and then for herpes it'll have some number after it as well. Or you've heard of the rabies virus, measles virus, Ebola virus, etc., etc., etc. Those are all the specific species. So this is going to kind of tell us a lot of information about that. Okay, so let's go back, check what else do we need to know. So we covered molecular components and all of those, and along the way we've compared the morphology and structure of a virus to a living cell. Living cells have cytoplasm, they have organelles, they have molecular components to replicate. They've got chemistry happening in their cytoplasm. Viruses have none of that. Viruses are bad news wrapped in a protein, and they have to hijack living cells, any kind of cell, in order to use metabolism, to use those life processes to replicate and move forward. There's attachments are extremely specific, and it's very important to understand the components of viruses so that we can target specific differences in order to make effective antivirals. Because if we are unable to target metabolism of viruses in order to make good antivirals, because our cells are the hosts. It's our cells' metabolism that we would be targeting that would damage our cells and would not affect the viruses. Okay. So I hope that this was informative and very helpful to you.